The Prix Galien is a welcome initiative to stimulate creative research and promote excellence. Barack Obama. The Galien Awards ceremony is considered the equivalent of the Oscars night for the innovators in the labs and awards every year the best pharmaceutical product, the best biotechnology product, the best medical technology, best digital health, best incubator accelerator, best startup.
This is the right event, on the right issue, at the right time.
I thank His Excellency, the President, for bringing Prix Galien to Africa, and I look forward to the day when we will celebrate an African winner of the Prix Galien.
I'm particularly grateful to receive this award.
The awards are among the highest honors in science and commerce because they lead to improvement in the human condition.
The Prix Galien Awards recognize the world's brightest minds and most innovative companies. They are a true celebration of the hard work required to produce life-changing interventions.
That is what makes us optimistic about the future.
Congratulations to all of you.
Make a difference. Join the Galien Foundation.
Welcome to the Galien Foundation Week of Innovation webinars here in New York. The objective of this week is to provide additional context and background for our largest annual event, the Prix Galien USA Awards, to be held this Thursday here in New York City. We've developed this new webinar series to give our nominees for Best Pharmaceutical, Best Biotech, Best Medtech, Best Digital Health Product, and Best Startup Accelerator Incubator category greater vitality and visibility in describing their journey of medical discovery. It's especially timely as the Galien Foundation marks its 50th Golden Jubilee year, represented by our Golden Jubilee Forum and Gala Awards dinner on Thursday. All told, we're recognizing the best of the best in biomedical innovations in these last 50 years. This year's Galien USA Awards has a record 147 nominees for 6 categories, coming from 126 companies, covering 15 diverse therapeutic areas, ranging from cancer, cardiology, and neuroscience to infectious and very rare genetic diseases and vaccines. We will be looking closely at all of these areas throughout the week. Today, in our Best MedTech category, we will be holding 3 webinars due to the large number, 24, products representing this important field. In the hour we have allotted, we will look at the 8 nominees in disease, diagnostics, and digital therapeutics. What we will cover today relates to the central question: what is MedTech innovation doing for patients with highly specialized needs for treatment? We will have some time for your audience questions today, and those questions can be forwarded to the moderator using the link provided on your screen. So let's move right to our discussion and start with some introductions. My name is Jessica Federer, and I will be moderating for today. I was previously the Chief Digital Officer for Bayer, and I'm now in venture capital as a managing partner at Supernode Ventures, investing in early stage companies. So for our panelists, I'd like to start the introductions with Dr. Chris Eagle. Would you please introduce yourself?
Thanks, Jessica. And hello, everybody. My name's Craig Eagle. I'm the Chief Medical Officer of Guardant, and Guardant is a company focused on oncology, focused on liquid biopsy oncology. I look forward to the discussion.
Thank you. Dr. Bella?
I'm Bella Dennis. I am a urologist by training and currently Vice President of Medical Affairs at Lantheus Medical Imaging. Lantheus is a legacy imaging company, but what we're going to be presenting to you today is the innovation in the diagnostics and in the diagnosis and management of prostate cancer.
Wonderful. Dr. Asim?
Thank you, Jessica. Thanks, committee, for having me here in this panel, prestigious panel. My background is in imaging biomarkers. I've been in prostate cancer for almost 20 years. I've been leading this group at Lantheus Medical Imaging for the application of artificial intelligence and how we can use the technology to enable physicians in PSMA at Therapeutics and Diagnostics. So really excited to be here. Thank you.
Wonderful. Dr. Alexey Elshin?
Thank you, Jessica, and it's a pleasure to be part of this panel. So my name is Alex Alshin. I'm a medical oncologist by training. For the last 5 years, have been at Natera, initially focused on our oncology business, most recently as the general manager of our early cancer detection business. Really looking forward to this discussion.
Thank you. Dr. Sangeeta Barhade.
I'm a pulmonary critical care physician by training and a transplant pulmonologist. And I'm currently the Chief Medical Officer in the Organ Health Business Unit at Natera, and I represent Prospera, which is a donor-derived cell-free DNA product to detect early rejection after transplantation.
Wonderful, thank you. Dr. Adam De La Zerda.
Hey, thanks so much for having me. So Adam De La Zerda, I'm the founder and CEO of Visby Medical. Also in another part of my life, I'm also the associate professor in the medical school at Stanford University. And here representing Visby Medical with our work over the last 8 years in infectious diseases.
Wonderful. It's great to have such a distinguished panel gathered here today. Thank you. So let's start with our first discussion question for the group. We'd like to talk about why your company chose to focus on the disease state and the indication of your product that's being nominated for the award. How are you approaching addressing this disease state? And tell us a little bit about the unmet medical need in the area that you're working in. So I'd like to start with Gardent, please. Craig?
Yeah, thanks, Jessica. So the disease area that we focused on, nobody really needs explanation for, is cancer. I mean, sadly, many of us are affected by cancer and certainly recognize on a personal level the unmet need, as well as then statistically, it's the second leading cause of death in the US. What we know is a sense of urgency physicians have in diagnosis, and currently cancer therapy is limited by access to tissue in a world of increasing understanding of genetics. So, for example, we know most patients in lung cancer, for example, are not getting genetic testing as prescribed by guidelines, in fact, over 80%. And so not only is there a time delay, there's also the biopsy issues that come with that assessment, and there's then also the accessibility. So what we focused on was solving these issues through our blood test type approach to try and understand and how to get results to patients faster, and also more accurately to get them onto the right treatment that gives them the precision of their genetic makeup of their cancer.
Thank you. I'd like to go to Lantheus next.
Thanks, Jessica. Following up on Craig's discussion about cancer, we too are focused on improving the care of patients with cancer, but specifically prostate cancer. As most know, prostate cancer in the US and worldwide is the leading solid cancer in men today, and is the second leading cause of death in men today. Despite huge advances in screening due to the widespread availability of PSA testing since the early 90s, and the improvements in surgical techniques with robotic approaches and improved radiation, the death rate or the mortality from prostate cancer, despite all these advances, has not changed in the last 40 years. Unlike other cancers, including breast, lung, colon, where patients are imaged, diagnosed, and managed, staged on the basis of molecular imaging or PET, or so-called PET imaging, prostate cancer has lagged far behind. And up until just last year, most men, and even today, most men have treatment decisions made based on standard so-called conventional anatomic imaging like CT scans, MRI, and bone scans. Those scans, unfortunately, are just not sensitive enough to detect disease in the early stage because they rely on anatomic changes in the body. PET scanning, on the other hand, detects disease at the cell level using molecular targeting. And, as I mentioned, breast, colon, lung, and most of the other cancers have been managed by patient access to PET scanners, with PET imaging agents, prostate cancer has lagged far behind until last year. In May of last year, the FDA approved the availability of F18 PSMA piflufolastat, better known as Pylarify, for commercial distribution and use. So Lantheus is very, very proud to have been the first commercially available PSMA agent for men with prostate cancer. We are focused in that disease because better imaging leads to better management, to better treatment decisions, and hopefully will translate into better outcomes.
Thank you. Asim, do you want to add to that?
Yeah, and just quickly, I think for us, innovation means quite a lot, especially for patients to bring about that change. And we have applied to this amazing PSMA that is revolutionizing prostate cancer restaging and detection, artificial intelligence to enable and give a tool for physicians to bring patient back into the conversation and understand the power of this imaging that Lantheus has brought to market. So we are very excited for that.
Wonderful. Thank you. Natera?
Sure. So I can kick it off. So at Natera, we're really focused on eradicating and mitigating human suffering through better understanding of genomics, especially circulating genomic biomarkers. And that's something, that we focus on across multiple business units, including our women's health business unit, oncology, and organ transplant. So just to focus on oncology initially, we set off to really tackle one of we think the bigger problems in medicine, and that's early stage cancer. So out of the 3 million Americans diagnosed with cancer each year, the actual majority of these patients are diagnosed with early stage cancer, where there is a chance of curing that patient if the cancer is caught early and appropriately treated. But for the last few decades, the big question on every patient's mind and oncologist's mind is who's really cured and who's not cured of their early stage cancer. And that's been very tough. And because of that, many patients have always worried, is their cancer going to come back? Am I getting the appropriate treatment after my surgery, for example? And can we actually pick up the recurrence early? And that's where we developed a test called Signatera to really address all of those questions. And Signatera was the first personalized and tumor-informed assay that was developed for this purpose. The way the test works is we actually look at a molecular fingerprint of the patient's cancer by sequencing the entire tumor that's usually removed as part of the surgery. We then develop a personalized assay to look for that fingerprint of that cancer in the patient's blood. And once created, this assay can be used longitudinally to ask the question, for example, is the cancer gone? If we don't find that signature after surgery, that means more likely than not that that cancer may be cured. But if we find that fingerprint, for example, after surgery or during surveillance, there's a high probability that cancer is still in the system, and appropriate steps can be taken to try to either eliminate that cancer at the earliest stage or try to pick up the recurrence at a stage that's most treatable.
Thank you, Alex. Sanjita, do you want to add to that?
Yeah, I'd like to echo Alex's comment that Natera is a leader in cell-free DNA technology. And with over 10 years of experience in this area, we are committed to improving patient care through innovation and scientific rigor across many clinical indications. And it's with this in mind that we chose to focus in the area of transplantation, as this is a life-saving procedure for patients with end organ failure. And importantly, this transplantation also improves quality of life in these patients. And it enables them to go back to their activities of daily living, going back to work, going back to their families, and being able to spend quality time with their families. When I was in practice as a transplant pulmonologist, I saw many patients who couldn't even leave their homes because they were so short of breath. And many of these patients have less than 2 years to live. Likewise, patients with end-stage kidney disease spend 3 days a week going to dialysis centers for up to 4 to 5 hours per time, and they also have a shortened lifespan. So transplantation can improve the longevity as well as the quality of life in these patients. Currently, there are over 40,000 transplants performed yearly in the US, and over 120 transplants performed worldwide. And this is increasing. In fact, in the US, there was a historic milestone, having performed 1 million organ transplants to date. So I wanted to mention that the unmet need in patients who have undergone transplant is a timely and accurate diagnosis of rejection of the transplanted organ, which we know can lead to organ failure and death in these patients. And unfortunately, over 30% of kidney transplants will have graft failure by 10 years, and this is related to rejection. And the statistics is even worse in lung transplant, where over 60% of patients will not make it to 10 years after lung transplant. Right now, the standard of care to detect rejection is to perform biopsies of the transplanted organ. We know that these biopsies are invasive. They have associated complications of bleeding and hemorrhage into the organ. They also can have life-threatening complications, including pneumothorax in heart and lung transplant, as well as heart arrhythmias in heart transplant. And as a transplant physician, I know that these biopsies are very subjective. And even with the most expert pathologists, they can have significant variability in calling rejection on the biopsy. These biopsies are taken from very small portions of the transplanted organ, and therefore they can be non-diagnostic at times. And then some patients are unable to tolerate these biopsies. So this is why at Medtech, we developed Prospera, which is a non-invasive circulating biomarker that measures donor-derived cell-free DNA in the plasma of transplanted patients. This assay utilizes over 13,000 informative and pan-ethnic single nucleotide polymorphisms, or SNPs, that are massively multiplexed by PCR and can identify rejection in the transplanted organ. Thank you.
Thank you. Let's go over to Visby. Adam?
Hey, thanks for having me. So I think maybe one of the common themes you're hearing in this diagnostics panel is kind of an underlying assumption of how medicine typically works. We usually tend to diagnose a patient and only then we treat them based on that diagnosis. For example, in the world of cancer, we first find out somebody has cancer, only then we consider giving them chemotherapy. You're never going to see a doctor telling a patient, I'm not quite sure if you have cancer or not, let's give you some chemo and see how you're doing, right? And that's really across the board with heart disease and diabetes, right? We first find out somebody has diabetes, only then we consider giving them insulin. And really, there's only one major exception to this rule in the world of medicine, where we treat you first and diagnose you later. And that's the world of infections. So think bacterial, viral, parasitic infections, whether that's an eye infection, an ear infection, a respiratory infection. COVID is a respiratory infection, but there are so many others. Urinary tract infections, sexually transmitted infections. So this kind of goes on and on. 1 in every 2 patients that walk into a doctor's office is there because of an infection. And yet our practice today is exactly going backwards, right? So we're kind of in the dark ages when it comes to dealing with patients with infections. And the interesting thing is the technology does exist to diagnose those patients properly. It's called PCR. We've all had probably multiple PCR tests over the last couple of years for COVID, but really across the board, PCR in a big instrument, typically something the size of a sofa, costs close to $1 million. It is incredibly accurate. If you collect a sample from the patient, send it to the lab somewhere in the dungeon of a hospital somewhere, there's this big PCR machine that's testing your sample. It's just that by the time the result comes back to the doctor, you're now no longer with that same doctor. You've maybe moved on, maybe you're somewhere in the hospital, maybe you're at home, anxiously waiting for your result. The doctor already decided how to treat you, whether that's to give you an antibiotic, an antiviral drug, or maybe just say, look, I don't really know how to treat you, so I'm just going to guess. Now, there's actually a name to this process called guessing. We call it giving somebody a presumptive treatment, which really is just a fancy way of saying, I don't really know what you have. So again, we're going to give it our best shot and see if it works or not. Now, in a world where bacteria become resistant to different antibiotics, that process of guessing is becoming less and less and less effective, to the point where just giving somebody this broad spectrum antibiotic is just not a good practice anymore. And so realizing that and realizing the fact that PCR does exist and it's very accurate, it's just not accessible to us when and where we need the test result, we asked ourselves at Bisbee now a little over 8 years ago, how can we make PCR a lot more accessible to anyone, anywhere, anytime? How can we take that big machine and miniaturize it to something that you can maybe hold in your hand, that not just your doctor will be able to run in the clinic or your pharmacist maybe when you go next time you go to the pharmacy, but can we even enable you, the patient, to test yourself, test your loved ones at the privacy and convenience of your own home? And so fast forward a few years, we actually did it, and it works really well. I brought it here to show you. So the device I'm holding here, if you can see my video feed, is the world's first and only single-use rapid PCR machine. There's nothing like this, and we just got FDA approval for this just a couple months ago. And what you can see here, it's our first product. We're looking for, in this case, sexually transmitted infections. This whole thing that I'm holding here is a single-use disposable, but still an actual PCR machine. The way that it works is you put the sample here where you see it says button 1. In the case of sexually transmitted infection, it's either a vaginal swab or in the future urine for males. Once you put the sample here, you close the lid and you press those 3 buttons, 1, 2, 3 in this order, and you are done. In less than 30 minutes, not hours or days, in less than 30 minutes, you'll get an answer right here. We can test for a number of infections. In this case, we can see we're looking for 3, chlamydia, gonorrhea, and trichomonas. Those 3 infections actually have identical symptoms, so the doctors can't tell them apart when they just look at you. But it's very clear you have one of them, but unfortunately, those 3 infections have very different antibiotic to give the patient. And so now for the first time, we can actually test and treat the patients while they're in front of the doctor, and hopefully soon enough, even allow patients at home to test themselves. And then through a very simple telemedicine session with a digital pharmacy that delivers drugs to your home, even allow patients to go from having symptoms to a diagnosis to a pill going into their mouth, and have this whole journey take no more than a couple of hours, and they would never have to leave home for that.
All right, Adam, thank you. We have some pretty incredible innovations to keep talking about today, and you brought in a nice segue, which is the reason we're all here, and that is the patients and also the clinicians that are supporting them. So my second question is to ask you all about what you've seen from the clinician and patient response so far. So what are the outcomes that you're seeing, and how are you hearing from patients and clinicians? What is their reaction to what you've worked so hard to innovate and bring to market? This time, let's start with Lantheus.
Well, thank you again, Jessica. And, I think that the recurrent theme that we've heard this morning, whether it's in transplantation, a diagnosis or assessment of treatment response to cancer, infections, is the notion of precision. And I think that, we all hear about precision medicine, and precision medicine is quite simply the right treatment for the right patient at the right time. And that's, across all these disease states. So in terms of prostate cancer management, the disappointment for men with prostate cancer has been that, despite, as I mentioned, major advances in surgical and radiation therapies and even medical therapies, 50% of men who undergo radical prostatectomy or radiation therapy with curative intent, 50% of those men will fail. And as Alex mentioned, it's important to be able to diagnose failure early, because that's your best shot at treatment. But much more important would be to prevent failure in the first place. In our studies, we've shown that in 30 to 35% of men, at the time they undergo radical prostatectomy, who have negative CT scans, negative bone scans, and are thought to have organ-confined disease and therefore consent to a procedure such as a radical prostatectomy, up to 35% of those patients on PSMA PET scanning will have disease outside the prostate. So given what we know now, it's no surprise that if 35% of men go into surgery with some evidence of disease outside of the prostate, that many of them will fail at a later date with recurrence of the PSA and require additional therapy. So, in terms, the patient reception has been huge. Asim and I were last week in Barcelona at the European Association of Nuclear Medicine meeting, and literally half the presentations at that prestigious meeting were on PSMA. Understanding PSMA, better utilization of PSMA, different PSMA ligands, and then more importantly, PSMA therapeutics. In other words, these are either antibodies, monoclonal antibodies, bispecific antibodies, or radioligands attached to PSMA that then bind to the cancer cell itself and selectively just kill the cancer cell without damaging any of the associated tissues. The reception, in terms of commercial success, has been huge at Lantheus. One of the sort of the good news, bad news things is that demand outstrips supply. And, some people think that's a good thing to have. On the other hand, if you're a patient waiting, access is important. So we are working to make sure that every patient with prostate cancer in the US has access to this new technology. I think that it will revolutionize our understanding of prostate cancer. The route of spread of prostate cancer, which we thought we had a good handle on before, is going to change dramatically. And I think ultimately, not only will treatments change, but outcomes will improve. Asim?
Yeah, it's very appropriate, Bella. And just to add a few points to this revolution that PSMA is causing, it's also important to understand from data science perspective and deep learning and artificial intelligence perspective, data is the holy grail, and managing data better can also bring patient back into the conversation, to my earlier point. And it's all about the patients. In prostate cancer, PSMA is leading to a revolution, but also PSMA is enabling new questions that can be asked that were not being asked before. In prostate cancer, for example, it's very difficult to quantify response to a treatment, to understand which treatment could work, so to understand avidity. With the help of a standardized objective technology that is enabled with artificial intelligence, we can answer these questions. We can enable physicians to ask if a treatment is working for a patient, so they are not exposed to unnecessary toxic events. A progression of a disease could be identified sooner than later. Still, in prostate cancer and guidelines, there is no way to assess response to a disease in the bone, because the bone, when the disease gets to the bone, unfortunately, is very sclerotic, very slow growing. So understanding response is very important in bone. So here again, data can play a very important role in understanding the power of new innovation that is coming in medicine. And that's where we are seeing the response from physicians has been tremendous, because they are getting patient back into the conversation. They are able to show the images and understand if their disease is responding to the innovative therapy that they're being exposed to. Yeah. So for us, it has been quite an exciting journey. We were the first technology that is AI enabled, that was cleared by FDA. And we are quite excited that we are continuously sort of breaking these bounds and enabling innovation to change how care management can be improved for patients.
Jessica, if I could add just one more point, a metric is that, you had asked about adoption and enthusiasm within the physician network and physicians in general. Within 6 weeks of approval of PYLARIFY by the FDA, the NCCN Prostate Cancer Guidelines Committee adopted it into their standard workup for patients at initial staging as well as recurrent. And I think that's record time for the NCCN.
Wow, thank you for sharing that, Bella. Let's move over to Natera. Alex and Sanjita, will you tell us a little bit about the patient and clinician response?
So I can kick it off. So for Signatera, we've seen, a very, very fast adoption since launching the assay. in 2019. And we believe that's because it really addresses some fundamental, outstanding issues in oncology, namely, is the cancer all gone and is it coming back? We believe that this is also driven by just a large amounts of data that's being generated. And we think prospective clinical utility data is first and foremost the most important type of results that can really show how tests can be used and how it can improve outcomes. And that's just to kind of highlight two studies that we're involved in. The first one is a big study being done both in Japan and the United States. In the United States, it's called the NRG 008, or the Circulate study. And the study is actually asking the question, well, can ctDNA guided or Signatera guided treatment for early stage colon cancer improve outcomes? And just to kind of walk through a patient's journey, for example, after surgery for an early stage colon cancer, the big question is, should we use adjuvant chemotherapy? And this is extremely toxic therapy given sometimes for up to 6 months. In a large percentage of patients, severe diarrhea develops, neuropathy develops to the point that patients can't even button their shirt. And some of these symptoms can be permanent. And there's also, unfortunately, a small risk of just dying from the therapy. And we know this therapy in aggregate only helps at most 1 in 10 patients. So finding that patient where this therapy is going to be effective is critical. So in this study, patients who are Signatera positive or have a liquid biopsy signal picked up in their blood are actually being randomized to either get more treatment or get the standard of care treatment. Really ask the question, can these high-risk patients benefit from more aggressive therapy and increase their chance of cure? But for patients who are Signatera negative after surgery, for the first time, patients are being randomized to either standard of care adjuvant therapy or complete observation. For the first time, really testing the ability to avoid using toxic therapy in patients who are not going to benefit from it. So this study is ongoing. We're excited to see the results. The second example I want to highlight is in breast cancer. Again, there's millions and millions of breast cancer survivors in the United States. And the big worry on many of these women's minds is my cancer coming back? So another study in a partnership with GSK that we're running, we're asking the question, if a woman becomes ctDNA positive during surveillance, and we've shown in other studies that our signal can precede recurrence by up to 2 years, does intervening early give one of these women, for example, a chance at second cure? So before the tumor actually comes back, if we can find that signal that the cancer is molecularly recurring, can we treat on that molecular recurrence and prevent recurrence in a subset of women? So all of these studies are ongoing, and we're really excited to await the results being generated.
Thank you. Sanjita?
Yeah, and likewise, Prospera has been very favorably looked at by both the clinicians and the patients. We know that Prospera is able to provide an accurate and very precise assessment of early graft rejection while avoiding these invasive, needless biopsies and their associated complications. So with that being said, we know that the use of Prospera eases patient anxiety and discomfort related to these biopsies. And sometimes up to 12 to 14 biopsies are done in the first year after transplant. And so I can tell you in the centers that have started to use Prospera, that number of biopsies have been cut in less than half of those that are usually required. So for patients, that's actually something that they're really, really looking forward to. In addition, Prospera improves access to healthcare. For those patients who live in rural areas or live a distance from transplant centers are unable to travel to the transplant center for getting a biopsy. And so they actually help to improve access to healthcare. So we have a large portfolio of clinical studies that support the performance of Prospera, including our clinical validation of Prospera in kidney, heart, and lung transplantation. And along with these studies, we have registry data for all of these different organs, which are very, very insightful in showing how patients actually respond very, very positively to this whole concept of not having to undergo so many frequent biopsies and still feeling comfortable that their organ is not rejecting. And And what's really important, what we're really proud of, is that we have two very rigorously designed randomized controlled trials in heart and lung transplant looking at Prospera as a diagnostic test for rejection, which is something that doesn't often happen in this field. So we're really, really proud of those two studies that are currently underway. We have 19 publications of Prospera in the clinical setting in all three of these organ transplants that show a very high performance, including a very high sensitivity and specificity in detecting rejection. And so with all of this being said, the clinician and patient response has been very favorable for Prospera. It's been widely implemented and clinically adopted across the majority of transplant centers in the US. And in the past year alone, 38,000 Prospera tests have been performed in over 200 transplant centers. So we're really excited about Prospera, and given the clinical importance of this unmet need in organ transplant, Prospera has gained CMS approval and is now Medicare covered. So
wonderful. Thank you, Sanjita. Over to Visby. Adam.
So as you know, I'm a medical scientist. I tend to measure a lot of things. So when you ask about patient impact, clinician impact, I tend to measure things. I'm going to give you a bit of a geeky answer, if that's okay. So we have now several hundred clinics around the country that already start using this Visby test as their new standard of care, which means that instead of collecting a sample and sending it to the lab, they can for the first time run the test themselves and tell the patient right there, right then, if they're positive and what they're positive for, and for the first time actually treat them based on knowing what the patient has, not just guessing what the patient might have. And one of the things we wanted to do is try to quantify and ask ourselves, what does it actually mean in terms of real impact to patients, to the clinical staff, to the overall healthcare system? And so we actually did a full clinical economic study, which we actually published not that long ago. And we just sent researchers over to those clinics, and we asked them to measure anything and everything they possibly can. So anything that has a unit of measure of either dollars, minutes, satisfaction levels, accuracy of prescription, anything that's measurable, we try to measure. And what we've been able to show there in that study that we published later is that not only we're saving quite a bit of money to the healthcare system, it is so incredibly ineffective today to just collect a sample and send it to the lab and make a guess and then chase down the patient later. But you're also saving a lot of time for the clinical staff. They no longer have to start chasing down patients. The problem is many times those patients are what we call lost to follow-up, lost to care. And in some of the settings, like in emergency departments, urgent care centers, STD clinics, sometimes as many as 40 or 50% of patients just don't pick up the phone later or don't come and take the medicine at the end. And so we've been able to kind of really try to measure how much time we're saving, how much money we're saving to the healthcare system, also what the satisfaction looks like, both for the clinicians as well as patients. Now that patients for the first time go into the clinic and actually get treated based on what they actually have, that's kind of a revolutionary concept for them. And also, how much did we improve accuracy of prescription? The accuracy of prescription is actually particularly interesting. What we've shown is using our test, you're reducing about 70% the prescription of antibiotics, again, that is completely unnecessary. Think about it, where the doctor tells the patient, I'm not sure if you have chlamydia, gonorrhea, or trichomonas. I'm just going to guess, let's treat you for chlamydia. Well, it turns out the patient had gonorrhea. And so using our test, you just save the need to give a prescription for chlamydia, which again, the patient does not have. You're not going to do any good giving this patient antibiotics. In fact, you're only exacerbating the problem of antibiotic resistance. And you also undertreated the patient for what they actually needed to be treated for, a good treatment of, in that particular case, of antibiotic called ceftriaxone. And so just across the board, improving the approach for how to deal with antibiotic resistance and reducing unnecessary prescriptions of antibiotics. But maybe I thought maybe to not to completely give you a geeky answer here, just share with you one anecdote. So I actually visited some of those clinics and had a chance to ask some of the nurses that actually run the test to give some real life examples of how this was received by patients. And so this nurse came to me and told me, in the training, you told us that it's all about finding the patients who are positive and then treating them so that by the time they leave, we treat them for what they have. She goes, I think you completely underestimated the value of telling somebody they're negative. And so she goes, let me tell you about this patient I had here just a few days ago. She came in, she was worried she might have a sexually transmitted infection. She didn't tell her husband she'd see her because she was worried he's cheating on her and she might have gotten this STD from him. In this one visit, I was able to tell her that she's negative, as in she is not having a sexually transmitted infection. And what she has there is something completely different. She goes, this patient gave me a huge hug, started crying, showed me a picture of her family with her kids, and she told me, you just saved this family. And she goes, don't underestimate the value of telling someone that they're negative, even though you guys are so focused on telling us that we should be treating those who are positive and the meaningful impact that we have on positive patients. Then I'll share just one quick anecdote as well.
Thank you, Adam. Craig, over to Guardant Health. Please tell us a little bit about what you're hearing from patients and from clinicians.
Yeah, so I think, we need to come back to a little bit the discussion we've been having around cancer. And, we know that cancer has been moving as a genetic disease, now understanding that genetics, and then the drugs that target that has been rapidly improving. The problem's been, of course, access to those treatments, either because of the lack of ability to get tissue biopsy, or the inability to run the molecular testing. And Guardant has attacked this problem by enabling a simple blood test, our product at Guardant 360, to actually go and take a blood test and enable the biopsy through the blood of the molecular basis of that disease and get to treatment. Part of the other issue, of course, is that timing of treatment. When you have cancer, time is of the essence. And so the blood test also enables a faster turnaround time compared to tissue. So as we develop the first of its kind liquid biopsy to solve these problems, really the question, the beauty about healthcare and innovation, as we're all here today, is the immense amount of knowledge and expertise in the community and the assessment of our technology as we move forward. And so if you look at the assessment of the technology and the publications as a measure, we have over 250 publications in peer-reviewed journals on the technology, for example, to demonstrate the ability of that technology. Currently, over 9,000 oncologists have been ordering our test, and we performed over 200,000 liquid biopsy tests on patients. And finally, of course, importantly, it's how to get it to the patients. And so what we end up with is actually having insurers covering over in the US, for example, as well as globally, but in the US, over 200 million lives now are covered for our test to get patients access to the test. And then similarly, we hear anecdote stories about the biopsy helping save somebody. So we know we've heard 50% of prostate cancer is in the bone and can't be biopsied easily. Breast cancer, a third of women with breast cancer have it in the bone and can't be biopsied easily. And so we often hear from a patient, and just the other day I heard from a patient who was discovered to have a potential biomarker in lung cancer that had been being treated with chemotherapy, and the target for that one was ALK. And that's a tablet, not chemotherapy, not injection, but just a tablet taken each day. And basically this patient had been having chemotherapy because they couldn't access the tissue and find the molecular target, and through a liquid biopsy test was able to simply go onto a tablet and had a spectacular response to that tablet and treatment. And the patient was extremely ecstatic, obviously, for this breakthrough. So these are the sorts of levels of impact we're having, and the acceptance through the community, whether it be guidelines, colleagues in oncology, whether it be the impact patients report to us, and ultimately the proof is in the pudding with the scientific review that goes with publications. So we're very delighted on how we've been able to bring part of the solution to cancer through this type of technology.
Thank you, Craig. So now as we move to the last question of our panel discussion today, I want to talk a little bit about the challenges of being among the first in your space to do things. So, in the drug discovery and development space, we have pretty defined processes of how you go from finding a molecule and getting it through the decades of development and investment into your medicine cabinet. But the spaces that you all operate in are relatively new. And as we all know, our industry is risk averse, and doing new things is very difficult. So I was wondering if you could each share some challenges or insights from your development process that might help other innovators in the space, encourage other innovators in the space, or even challenge regulators to look at things differently. For this question, I'll start with Natera, and maybe Sanjita, if you'd like to go first.
Sure. So the challenges of bringing any product from proof of concept and ultimately to the clinical setting, I believe, are time and resources in order to do this in a robust scientific manner. So when we conceived of Prospera, we went through a very rigorous process of analytical validation. We wanted to make sure that the test was reproducible, it was precise, it was accurate. We then went through our clinical validation studies, and of course, importantly, looking at clinical utility in the healthcare setting to increase adoption and go to market authorization and reimbursement. But despite this evidence, there always remains the challenge of increasing physician adoption when there's a new diagnostic test or medical device. And I don't think this is unique to Prospera. I think that this is probably what we're all going to face as we start to bring new innovations into the healthcare setting. And so as we approach this, we think about the changing physician behavior as really a multi-pronged approach, which is really based upon a belief in the test, that's based upon all the clinical evidence that you accumulate. But there has to be continued awareness for the physician as well as the patient, and continued education of the test's clinical utility. And in our setting, we can't forget that transplant biopsies have been the mainstay or the standard of care for so many years. And so despite the strong performance and the non-invasiveness of Prospera in comparison, it's still not seamless to change physician behavior. But, I think we're working towards it. And, Prospera is a lab-developed test. It is both CLIA and CAP certified and has recently gained CMS coverage. And so our focus today is to continue to increase awareness and really educate both the clinician and the patient of this Prospera test in the world of transplantation.
Thank you, Sanjita. Alex, over to you.
Right. So I think whenever you're first, that always comes with a host of its own challenges. And the way I kind of like to think about it is three large buckets. The first one is education, both of physicians and patients. The second one is regulatory. So how does a new device get regulated? And the third one is always access and reimbursement. So maybe kind of starting with the first point, education. So with Signatera being the first molecular minimal residual disease test, this has really flipped a lot of oncology on its head. For example, how we stage cancer is slowly starting to change. We've traditionally thought of cancer as being stage 1, stage 2, stage 3, stage 4. But many of our studies, we've shown that a stage 1 patient who's CTA positive is going to have a markedly worse outcome than a stage 3 patient who is Signatera negative. So this is now forced the community to actually rethink staging. And there's actually a concept now called TNMB staging. And TNM staging is traditional staging, but TNMB adds a B component, and that's blood-based MRD status. So you may in the future actually stage patients to say, the tumor is this big, the lymph nodes are negative, but the patient's MRD or Signatera signal is positive. And because of that, this patient should be treated this way or that way. So for example, working through something like that has been challenging and has required quite a bit of education and also lots and lots of high quality data. And I think because of that, we continue to publish, we continue to run large prospective studies. And surely, over time, all of that data adds up to start changing the perception of these tests and get more physicians using them to really improve patients' outcomes. The second one is regulatory. So I think whenever there's something new that doesn't have a precedent, it's really hard to think about how something like this should be regulated. So Signatera right now is a lab-developed test regulated through the CLIA/CAP pathway. But we're really committed to working with the FDA to get the test FDA approved. We're actually doing that by running large prospective pharmaceutical sponsored studies where Signatera is being used as a companion diagnostic to some of the largest, kind of most popular drug classes that are being utilized in cancer therapy today. For the first time, this has now allowed these therapies, some of which Craig has actually mentioned, to come into earlier stage of cancer. So not just being available for patients with metastatic or advanced disease, but with the help of tests like Signatera, really be able to be used in the earliest phases of cancer therapy. And then the last one is access and reimbursement. And I think with diagnostics and many medical devices, the pathway sometimes cannot be not as clear as in other settings. So I think here we're working with Medicare, we're working with private payers, and we're working with patient advocacy groups as well to really think, how can we allow every patient who can benefit from Signatera to have access to it?
Thank you, Alex. Adam, tell us a little bit about the challenges of bringing your new test and device to market.
So a few years ago, when my daughter was 1.5 years old, we ended up in the emergency department at Stanford. She was having difficulty breathing. She wasn't doing very well. And they immediately brought up a bunch of ENT doctors that recommended us that they should go into her lungs because they were suspicious that she might have inhaled a piece of toy, maybe maybe a coin or something into her lungs. So they said, if we don't take it out very, very quickly, if it's there, which we're not sure if it's there or not, but if it's there, if we don't take it out quickly, she might develop pneumonia and then die within hours. She ended up, in the hospital for quite a lengthy hospitalization. It was a terrible, terrible experience. And it actually took 24 hours from the moment we walked into the hospital for the lab result to finally come back and tell us that actually she has nothing in her lungs, at least not a piece of toy as they initially suspected. But what she had was a virus called RSV, which is not a fun virus to have, especially if you're 1.5-year-old, but it's certainly not life-threatening in that same way that they thought. And that whole journey with so many radiation to her lungs, with x-rays and so on, and of course them trying to look inside, we could have completely been avoided. And I remember that's quite a few years ago now, thinking to myself, boy, if they only had one of those tests like the ones we're developing for that virus, of course, at the time we didn't have that product on the market, but that if they just had this test, all of that could have been saved. It makes so much sense. So you always start the development process with either a personal journey or maybe a friend or someone that you look and say, it makes no sense. Someone has to do something about this. Clearly, I'm not just the only one experiencing this. There are many, many millions of people around the world suffering because we're treating before we're diagnosing those poor patients. Now, the interesting thing, and maybe kind of the woken up moment for me was, fast forward a few years, the technology now does exist, and we're now having hundreds of clinics around the country use this. But it's so much more than just, in terms of the development process, so much more than just coming up with a solution to a problem that we can all agree has to be solved. And just maybe to pick on the one thing in particular, as you pointed out, doctors tend to be a little slow in adopting tests or generally adopting new technology. And it's just incredible to me to understand and realize just how much incentive drive behavior. To what extent you really need to think about all the stakeholders. We call it the 3 Ps, the patient, the provider, and the payer. And how do we make sure that all 3 of them truly align their interests and want the same thing for the benefit of the patient? So you really have to think about this test of mine. Is it not just helping the patient, but is it also making the doctor's life simpler? Is it saving them time? Is it to make them more money, perhaps? And how does the insurance company think about this? Is this now going to cost them a lot more money? Because if it does, boy, that's an uphill battle. And I think diagnostics generally is certainly in a much more difficult position than therapeutics, just because in a diagnostic world, the patient was sick before our diagnostic. The patient is still sick after our diagnostic. Now we hope that delta information we just provided you is worthy enough to justify kind of the maybe additional cost or maybe some saving downstream costs. But if you think about the ultimate impact it could have, maybe 5% of our overall healthcare costs are diagnostics, but they control downstream about 85% of the overall healthcare costs. So the leverage is huge if you could just align the interests properly and make sure that we don't just have the right technology, but also give it to the right people and with the right set of incentives around.
I'm gonna jump in and ask, because we're getting a little close to the end of our webinar. I want to make sure Craig and Bella and Nassim also answered. Craig, over to you at Guardant.
Yeah, thank you. And I think us all here at the Galien panel here and all of us in the audience have demonstrated one of the things that I think is most important for innovation success. You know, we hear about education and barriers around pricing and payers, and technology obviously is an important thing as you develop technology. But really for us in the scientific world, of course, we're taught to doubt everything. You know, this is often translated into the null hypothesis for statistics, nothing works. And so really, I think the biggest factor for success in innovation, certainly in Guardian's case, is the actual people involved. You need people that are dedicated, that see an unmet need. We heard from Adam in the case of his daughter, and basically have a passion against this wave of null hypothesis that myself as a science physician is trained to doubt everything. And I think the panelists here have all shown that with all their technologies, along with the entire team at Guardian, to bring this type of technology through a sea of industry requires passion, dedication, and commitment, with that end goal that you're really bringing something special to healthcare and something that really makes a difference. And so for me, the rest is just tactics now, whether it be getting FDA approval in our case, or whether it be getting payers. These are all obstacles to overcome, but without a passionate team, it's really impossible. So probably that's how I'll come at this innovation question.
Thank you, Craig. The last 2 minutes, Bella and Nassim.
Well, I'll try to be very brief. I mean, you know, Alex highlighted very exactly the challenges for all of these products. One is education, the regulatory pathway, and then access, which depends to a large degree on adoption within guidelines, reimbursement by Medicare and commercial payers. But the one challenge that's very unique in the space of nuclear medicine is production, because it's not like a drug that you take off the shelf. These agents, Clarify is an F-18 labeled agent, so it's labeled with a radioactive compound, which has a rapid decay. In the case of Clarify, the half-life is 110 minutes, so it's just under 2 hours. So we had to establish a network of cyclotrons around the country that are able to make this product every day to meet the demand. And if you can imagine treating thousands of patients daily or imaging thousands of patients daily with cyclotrons running day and night throughout the country, and getting those doses to the right imaging center, to the right patient at the right time, it's quite a challenge. And then we met that head on and have been quite successful in doing that. In terms of development, Lantheus acquired the product from Progenics Pharmaceuticals, who acquired it or licensed it from Johns Hopkins. So we were not hands-on, involved in the development and the approval process of the product. But I will turn it on to Asim, because he's been involved in the development of the artificial intelligence component from day one.
Yeah, talk about challenges. Sometimes really feel that being very interesting if I would have chosen a field to be in social media and like my cat videos. It's like, our field is full of challenges, and I would like to echo what Alex and Sangeeta said, and also Craig. People, regulatory challenges, analytical validation, clinical validation, and ultimately market access and adoption. One of the unique things that we are facing in artificial intelligence in healthcare application is the important that people often forget, the ethical responsibility of a deep learning neural network that needs to be unbiased. There's also a challenge of diversity of data. We want to make sure that the data that AI is learning is not skewed or biased in any way, just like any humans could be. So for us, new innovation really brings these unique challenges, but it takes passionate people, committed folks to actually take that to fruition through the rigor and bring it to the market. And it's not easy, but it's very rewarding, because when you see the patients impact, the patients smile on their faces when they see the image, when they see the score, and they ask the question, oh, what doctor, this means that my disease is improving, right? And that's great. And that's so rewarding than anything else. So challenges are really important in this field. And we are excited
Indeed.
to be one of the ones. Yeah. Solving
Well,
those.
you have all brought your passion and your perseverance, and we have to wrap up this session so the next one can begin. But thank you all for joining us today. This webcast will be available on the Galien Foundation's homepage, and we look forward to seeing you Thursday at the Jubilee Forum. Bye-bye.
Thank you.
Thank you.
Bye-bye.